Alright, Assalamu alaikum. I'm your teacher here, Anas Faruqi, and I will be teaching you Cambridge O-Level Biology. And today we will be discussing cell structure and organism. The book I'll be referring to is Cambridge O-Level Biology by D.J. Meckin. This book has been referred to by Cambridge themselves on their website, so you could refer to it as well. And throughout the course, I'll be using this. Alright, at least for the series of O-Level Biology. And I'll be breaking down each and every concept in the simplest form so you guys can understand it, and then you guys can attempt the questions without any hurdles. And I'll be trying my best to make these concepts very crystal clear so you guys don't have any issue understanding them. Now, moving to our first topic, which is cell structure and organization. First, we need to know what is a cell, right? The basic definition of what is a cell. Cell is basically a functional unit of life, right? And a functional unit of life is basically, it means that this is the requirement of the organism to exist, a cell, right? So, under the microscope, the cell would look like a tiny box like structure, right? And that's what they have stated here as well. Under the microscope, it would look like tiny box-like structures, and they are called cells. And cells have multiple functions. Each cell has their different types of functions. Like for example, the cells in your heart have their own different type of function, whereas the cells in your kidneys have different type of functions. So all of these organs are made up of cells and then tissues and so on and so forth. We'll be discussing the lineage of these structures further on, but to just break it down into simple words, cells are the functional unit of life, and they are box like structures, tiny box like structures under the microscope. Here we have a cross section, a microscopic slide of a plant shoot on this image. And if you could see here, there's like box like structures present here, and these are cells. These are, each one of them is a different cell making up the whole plant, making up the whole plant stem, right? This is just an image. Now before we get into cells and different types of cells, one thing very important you guys need to know is how do we look at any sort of specimen, any sort of structure, uh, any sort of substance under a microscope, right? Something which cannot be seen from your naked eye, which needs a magnification on it, right? Now before doing that, first how do we prepare that sample, right? Number one thing which we need to know is how to prepare that sample is we soak it in melted wax, right? Here as they say, they soak it in melted wax, and then we let the wax get solidified. And so basically the reason why we do this is whenever we have any sort of tissue or any sort of content, the reason why we cannot just slice it in, make a cut out of it with a razor blade, is because there are other problems that come with it. For example, if your specimen is very fluidy like structure or is easily breakable, so you will not get a proper sample of it unless you solidify it, and then you can cut that section easily. So to make that process easier and to keep the contents of it firm in their place, we solidify it by using melted wax, and then we use a razor blade to cut it. And as they said here, animal structures is more difficult to cut because they are most soft and flexible. That's why and like skin, muscle, that's why they must be soaked in melted wax. Now once they're soaked in melted wax, and once now you're using the razor blade to cut it, now there are two techniques that we use here to make a cross section, a cutting section of not a cross section, a cutting section to be precise, a cutting section of the specimen. For example, over here we have a plant stem. There are two ways they have shown. One is a transverse section, which is basically an horizontal section, and one is a longitudinal section, which means along its length, right? Along its lengthwise is a longitudinal section, and across its length is a transverse section. Okay, and As you can see here, the razor blade is being cut in this direction, horizontal, and when you do cut it, you will see this area of contents. This is the image you will see, right? If I make a cut. Whereas if I make a cut longitudinal, then I would be seeing the contents from this point of view, right? And that point of view is basically this filament is of course traveling down, or whatever it may be. I think it's an xylem. It would be traveling down like that. All right. So here, as they have cut the longitudinal section, you can see this is in its length, whereas the transverse section you will not see its length, you'll just see the top surface area, right? So these are the two methods of taking the section. It's longitudinal section and transverse section. All right, here's a picture of kidney tubules and how they may look, and you can see that these type of tubules look a bit different from these type, because all of them are their direction is different in forms, right? It's not necessary. For example, there's a plant stem over here. There could be another plant stem next to it which could be horizontal, and now even if I take a transverse section along both of them, although on the first plant stem I will be getting this sort of view over here, right? Whereas if I take a section of this, I'll be getting the lengths, right? So we also need to keep in mind that which direction is the specimen faced in. It's not necessary all the contents in the specimen are facing in the same direction, so you can get different views and different angles of that, right? So moving on to a few more important things is now let's move on to cells, right? Cells are, they have their own different sizes, structures, and their functions, right? And if you see, each cell is designed in such a way that it is complementary with its function. For example, if there's a structure which needs high level of absorption, there will be a thin membrane, right? It's because a thin membrane will let a lot of things absorb, whereas if there's a thick membrane, less contents will be absorbed into it. So that's one of the ways you can even identify cells, and their identification points is through how they look like their size or their shape, which help in their functional qualities. Now, a few important structures present within a cell, within its boundaries, and one of the most common structures are, one of the one is cell membrane. It's basically an outer boundary of the cell. It bounds the cells and its contents inside the cell. And the second is cytoplasm. Cytoplasm is a jelly-like structure which contains different types of organelles, different types of oil droplets for energy sources. And what are organelles? Organelles are basically different type of structural components of the cell within the cytoplasm, which help in different types of functions of the cell. For example, there are, we all know mitochondria. I know you all know that mitochondria is the powerhouse of the cell. It basically provides energy to the cell. So you will find that there are mitochondria that are going to be present in the cytoplasm. So this mitochondria has its function to produce energy. So similarly, there are multiple other organelles which have their own function and which help the cell grow, reproduce, and perform its activities within the cell. And of course, one of the most important things present in a cell is a nucleus. The nucleus is very important because it contains the genetic material. The genetic material which helps it to reproduce and replicate into multiple other cells. Here, as we can see, the nucleus is comparatively darkly stained comparatively to the cytoplasm. Cytoplasm, as we can see here, is lightly stained, whereas the nucleus, on the other hand, is pretty darkly stained. So this helps us identify what the nucleus is. We can even see there's mitochondria over here. Right now, the magnification on this image is very low. That's why you cannot properly identify the mitochondria. But you can see that the mitochondria and different type of granules and different other organelles are present within the cytoplasm, which help in the functions of the cell. All right, so moving on to what is cytoplasm. As we discussed earlier, it's a thick liquid, and it has food reserves like oil droplets, like granules basically, which contain starch. For what? For energy purposes, for helping in its functions and metabolic activities. It also contains organelles such as mitochondria for special functions. As I said, mitochondria helps in producing energy for the cell. So each type of organelles have their own sort of function. And 90% of the cytoplasm is made up of salts, sugars, and water basically. Water is one of the main contents of cytoplasm, and it other few minute contents such as salt, sugar dissolved in that water, right? More things included in the cytoplasm is lipids and proteins. These lipids and proteins are required for different functions of the cell, such as the lipids help in making the cell membrane. As we described earlier, the cell membrane is a boundary of the cell, but it's also made up of something, right? And to help make it in the making of the cell membrane, lipids are used, and even proteins, both of them. And I'm sure, as you all know, and you're studying in chemistry, that what catalysts are, they help reducing the activation energy, they help increasing the activity of the reaction, they help in decreasing the time of the reaction, increase the rate of reaction, to be very precise. Enzymes are basically biological catalysts, right? They're present inside your body, and they help in increasing the rate of reactions inside your body, and they're made up of proteins, right? So these are a few important points that you need to keep in mind. Now, cell membrane is a thin layer of cytoplasm. It's basically continuous with the cytoplasm. The cytoplasm, as you can see here, is continuous with the cell membrane. And one of the main functions of the cell membrane is that it controls whatever is entering into the cell and whatever is leaving the cell. It's basically एक पुलिस की तरह है कि वो देख रहा है कि कौन सी चीज़ें अंदर आने की ज़रूरत है, एंड वो देख रहा है कि कौन सी चीज़ें बाहर जाने की ज़रूरत है, बाहर भेजने की ज़रूरत है. तो बस पुलिस की तरह है, एंड इट्स टाइप ऑफ अ बैरियर. So that's one of its main functions, that it stops cell contents from escaping and controls which substances can enter the cell and leave the cell. This is a very, they ask you in exams what is the function of cell membrane, and this is very, very, very important. Structures such as what they could ask, what structures does it stop or let give out? Like, for example, it takes in oxygen, takes in food. Why? Because it requires energy, it requires oxygen to make energy, it requires water as well. And waste products such as what is, of course, if it's doing respiration, performing respiration, there's carbon dioxide production, and hence these wasteful products are excreted out of the cell. Now, moving on to nucleus, plural nuclei. And most of the nucleus are round like structures, as we have seen in the previous image. They have their own membrane as well. That's something to keep in mind. That cell has its one cell membrane, but the nucleus as well has its own membrane inside too. So what you need to keep in mind, if this cell membrane is controlling what is leaving the cell, what is entering the cell, then in some ways and in some forms, the membrane of the nucleus also controls what enters into the nucleus and what leaves the nucleus and enters the cytoplasm. तो ये भी बहुत important चीज़ है. Okay. So so whenever you look at the nucleus under the microscope, you'll see that it's darkly stained, as we also discussed that earlier, compared to the cytoplasm. And that's because it contains one thing very important is that it contains chromosomes. Chromosomes are basically made up of DNA, and they're proteinous in structure, right? And that's why they stain darkly stained under the microscope, because they have chromosomes, and chromosomes contain DNA. And one of the main functions of the nucleus is that it controls the cell division, cell replication or cell division. Basically, the cell producing more cells from its own cell, like the cell replicating into 2 cells, 3 cells, 4 cells, and so on and so forth. This is done by the help of nucleus. And one thing you need to note here, a cell without a nucleus cannot reproduce inside, so it cannot reproduce. And you will realize that further on in the chapter we will see that RBCs are one of those cells which do not have their nucleus, and hence if in the exam they ask you which type of cells cannot reproduce, you can give them an example of RBCs. Why? And why is that? Because they do not have a nucleus, and nucleus is required for cell division. And so basically this is one of the functions. One of the secondary functions as well is the nucleus controls what the cell is going to become in the future, whether the cell will become a blood cell or the liver cell, muscle cell. These all types of cells are different and act different, they look different, their functions are different. And who decides what the cell is going to become? It's the nucleus. The nucleus decides what the cell is going to become. All right, here's an image of how the cell replicates. First, when the animal cell is about to replicate, the nucleus divides. So they see it's replicated. The nucleus material has been replicated. There's one whole pair of nucleus, and then once the division of this nucleus has finished, then the cell starts dividing. And as you can see, the two daughter cells, the two daughter cells, they separate from in between, and they share this. They pinch from the cytoplasm. As you can see, it's a type of a pinch. And lastly, you can see that two cells are formed from one. So this is basically a type of cell division in animal cell. Now moving on to plant cells. Here we can see that they have given an image of two palisade cells of plant cell, right? As soon as you see this image, you find that there are two new things present comparatively to the animal cells. In fact, there's a few different things that you can point out. Number one being that there's another boundary outside the cell. It's called cell wall. No doubt that there's a cell membrane over here. Cell membrane is still present. Don't get me wrong, cell membrane is there. But now there's an outer boundary called the cell wall. All right. Now we also have here green type like structures called the chloroplast. This is something that was not present in the animal cell either. So here we have the chloroplast, and we will discuss about it further on in details. Secondly, we see here that there's a large vacuole, and this vacuole is pushing all of this cytoplasm, all of this orange cytoplasm that they have given here, to the periphery. Whereas in the animal cell above, as you can see, the cytoplasm was around the cell. It was not in the periphery. The vacuole was not pushing it around. So the vacuole here is like far greater and larger in a plant cell. And you can see that the nucleus has been pushed to the periphery, and not only the nucleus, the cytoplasm has been pushed to the periphery as well, because of such great content inside the vacuole, right? As we already discussed earlier, the nucleus has a nuclear membrane around it as well. Very important. Now let's discuss about plant cell. One of the main differences they have stated here is that it contains a cell wall, as we discussed, chloroplast, and a sap vacuole. The cell wall is made up of cellulose, a very important content. It's a carbohydrate, and which helps in the formation of the cell wall. They ask you this in exams as well. It's a non-living type structure. It allows water and dissolved substances to pass through it. So one thing that I forgot to mention here about the cell membrane is that yes, as it acts as a police, वो देखता है कि कौन चीज़ें लीव होनी है, कौन सी चीज़ें आनी है. तो इसका एक अल्फाज है, there's a word to this, that it's partially permeable, right? Very important. They can ask you what cell membrane is. Cell membrane is partially permeable. It allows a few contents to leave the cell, and it allows few contents to enter inside the cell. So whereas the cell wall here, which is present in the plant cell, it is not partially permeable. It is freely permeable. It allows each and everything to enter into the cell wall and each and everything to leave the cell wall. So there is no such selective mechanism for the cell wall in itself. And So that's almost the main part you need to know. Number one, that it's made up of cellulose. Number two, that's a non-living type structure which allows all the substances to pass in and out of it, and it's not selective like the cell membrane. um, as they have also stated, under the microscope, the cell wall is easily identified because it's one more structure outside. And in plant cell, the cell membrane is comparatively difficult to identify as it's very thin. But since the cell wall is so thick, it's comparatively very easier to identify. So moving on to vacuole, its large fluid-filled space is called vacuole. The vacuole contains mainly watery solution, sugars, salts. Mainly it contains water. And you could also call the vacuole sap or just vacuole. Those both words are correct as well. And as we discussed, it pushes the cytoplasm outwards. You can see this orange color cytoplasm which is present here is being pushed outwards. It's not in the middle anymore. It's being pushed outwards. Why? Because there's so much fluid content inside the vacuole कि वो जो सारा साइटोप्लाज्म है ना उसको बाहर की तरफ पुश करते जा रहा है. ठीक है. Now one more thing that we need to know here is जो एनिमल सेल्स है ना उनमें भी वैक्यूल्स होते हैं, लेकिन वैक्यूल्स इतने लार्ज नहीं होते. वो स्मॉल ड्रॉपलेट टाइप वैक्यूल्स होते हैं, राइट. स्मॉल ड्रॉपलेट टाइप वैक्यूल्स होते हैं. एंड वो एक टेम्पररी टाइम के लिए होता है. जब तक सेल को ज़रूरत होती है वैक्यूल्स की वो यूज़ करता है, एंड जब नहीं होती फिर वो डिग्रेड हो जाते हैं वैक्यूल्स. एंड वही चीज़ उन्होंने यहाँ स्टेट की है कि वैक्यूल्स आर नॉट परमानेंट एंड दे ओनली प्रोड्यूस फॉर स्पेशल जॉब. There's any sort of special function that the cell requires in that certain moment of time, those vacuoles are produced. And once that function is over, done with, or finished, then they destroy the vacuoles. One important content of the plant cell, as we see, is chloroplast. Chloroplast they contain green pigment like structures called chlorophyll. And what does chlorophyll do? Chlorophyll helps in trapping sunlight. It helps in trapping / absorbing sunlight. And with the help of that, as we know, plants perform photosynthesis. Photosynthesis is done by the help of sunlight. So the way that sunlight is absorbed is by the help of chlorophyll. Chlorophyll helps in trapping of the sunlight. Very important for the use of photosynthesis. As we discussed earlier, they have given two cut sections of a plant cell. As we discussed earlier, there's two ways. One is a horizontal, or you could say a transverse section, and one is a longitudinal section, right? Now, if I take a transverse section out of here, right, I would be getting something like this. You see over here, I would be getting an image something like this, and that's exactly what they have shown here. Whereas if I cut it longitudinally, it would be something like this, as they have given over here. If I were to make a longitudinal cut, then I would basically be getting something like this, which they have shown already. So I hope you get the point. This is a great example that they have used here, which can help you complete your concept. And they have told you they are three-dimensional, may have seen different shapes depending on the direction. So it's not necessary that you will see all of them parallel to each other, right? These two are parallel to each other, but that's not necessary. These palisade mesophils are parallel to each other, but that's not necessary, right? There could be another palisade mesophil right next to them, which could be horizontal. And when you make a cross section, it could have a different type of image being shown according to the way it's directed. You have to keep that in mind too. All right. Yes, now this table is really very high yield. You have to read it and you have to understand it. I will go through it. It's already, we have discussed most of it, but we're just going to have a review here. Cytoplasm, jelly-like substance with particles, organelles. As we said, there's organelles within it, enclosed within cell membrane. Which type of organelles? There's mitochondria, we already discussed that. There's nucleus, and it's a site for chemical reactions. Cell membrane, they have finally stated here that it's partially permeable. I already told you what partially permeable means. And it prevents cell from contents from entering or escaping the cell. It's a type of police mechanism. It sees what should enter, what should leave the cell. Nucleus containing the genetic material contains genetic material, which basically contains DNA. Genetic material in the contain, which is basically DNA, right? And in the form of chromosomes. And that nucleus basically helps in cell division. We already did that. Cell development, what the cell will become in future, that's called cell development. And cell activities, whatever the activities are taking place in the cytoplasm, it basically is a control center. We also know that mitochondria is a powerhouse of the cell, and there's a reason to that. I'm going to explain you why. Secondly, we call the nucleus also as a control center because it's controlling all the types of activities that's taking place within the cell, and it is controlling them, right? We have the mitochondria here. It's under the microscope you'll see it's circular, oval shaped, slipper shaped as well, and the main function of it is that is responsible for aerobic respiration. I'm sure you're familiar with what respiration is in itself. It's what the cells do to produce energy, right? To live, to reproduce, to grow, and all that. But what is aerobic? Now, what you need to realize here is aerobic means in the presence of oxygen. Mitochondria only performs aerobic respiration. It means it only performs respiration when there is sufficient oxygen present within the cell. And once there is not sufficient oxygen present within the cell, for example, when you're exercising and when you're out of breath, right? Like when you are out of breath, and so there's not sufficient oxygen at that moment. That moment, mitochondria are not functioning as much. And why is that? Because oxygen ही नहीं है. When जब oxygen ही नहीं है तो वो अपना aerobic respiration कैसे करेगा? And the respiration which is done in the absence of oxygen is called anaerobic respiration. It's called anaerobic respiration, right? And we'll discuss about aerobic, anaerobic respiration in further details in later chapters, but this is the basic thing that you need to know about this. And the reason why it's called the powerhouse of the cell is because it produces energy with the help of aerobic respiration. We have ribosomes as well in the cytoplasm. They're in two forms. Either they're bounded to some sort of membrane, or either they're free, right? Number one, they're bounded to a membrane. Number two, they're free. And the main function is protein synthesis. They help in synthesizing proteins, and we already discussed the uses of protein. Number one, it could be used to form cell membrane. Number two, it could be used to form enzymes. Until now, this is what the chapter has discussed with us. So the ribosomes help in synthesis of the protein. Cell wall is a tough non-living material made up of cellulose. Very important. They ask in exams what the cell wall is made up of. You need to know that. It's outside of the plant cells. So it has two functions. Number one being it prevents cell from bursting. For example, what if a lot of water moves into the cell, right? Water can move into the cell and move into the vacuole, and hence the vacuole pushing the cell out and out. The cell can burst, right? This is not safe for the cell. Now the cell wall, what it does here, it is so rigid, it is so strong that it does not let the cell burst, and hence it prevents the cell from bursting. It's very rigid and strong and gives structure to the cell. You can say the one is prevents the cell from bursting. Number two, gives structure and support to the cell, right? And a thing that they have discussed here, that it's freely permeable because it's not like the cell membrane. Vacuoles are fluid filled surrounded by membrane, contains salt, sugar. One of the main contents is water inside, and helps to keep the plant cells firm. As we have discussed, the plant cells, the cytoplasm and everything is moved to the periphery because of the vacuole, and it helps to keep the whole cell firm and strong, right? Lastly, chloroplast, organelle containing chlorophyll. Chlorophyll is what? It's a green pigment. It's a green pigment. inside the cytoplasm of the cell, and it helps in photosynthesis by trapping the light for energy. Now, under the microscope, with a low magnification, what we saw before here was here. We did not really see all the type of organelles present within the cytoplasm, and there's a reason behind that. That's because this is not at a high level of magnification. So if you look at the cell under a high level of magnification, you will realize now you would be able to see the organelles and more of the contents much more visible. So as they have stated, at a higher magnification, the cell no longer looks structureless, and you can see a few organelles such as ribosomes, mitochondria. As they have discussed, mitochondria here has an inner membrane and an outer membrane. Very important. In exams, they could ask you how many membranes are there in a mitochondria. It's double layered membrane containing an outer membrane and an inner membrane, and that inner membrane is inward pointing folds. I'll show you what they mean by that. Here you could look at a mitochondria. Number one, it's like a slipper shaped, or you could say oval or round shape like structure. It has an outer membrane, and lastly there's a inner membrane with folds, right? There's an inner membrane with its own type of folds. They're pointy folds here, as we can see here. There's not just one. I can give you more examples over here as well. You could see this is rod-like structure, outer membrane, inner membrane with its folds, and over here again another mitochondria over here. So I hope you understand, and I hope you can identify what a mitochondria may look like on an image photograph. So as we discussed earlier, they help in aerobic respiration to produce energy for the cell. All right, so this is the main things that we have discussed, and this is the animals. This is an animal cell because they said a liver cell. Liver cell is present in an animal cell. All right. Whereas if they would have given a plant cell, you would have seen chloroplast and a cell wall as well, which is not here because it's an animal cell. Now moving on, these are a few images, pictures, which is not that important. Yeah, now test yourself. Now, you know what? Let's test all of the knowledge we had. Let's have a bit of review. So, you are confident about what you have learned today. What structures are usually present in both animal and plant cell? Very easy. Number one being cell membrane. Let's start from outside. Cell membrane. Number two being cytoplasm. Number three being nucleus. Very good. Very easy. Now the structures that are present in plant cells but not in animal cells, that's also very easy. Now we start from outside again. Cell wall, and chloroplast. Which cell structure is mainly responsible for controlling the entry and exit of the cell? Very easy. As I said, the police and the structure that controls what enters and leaves the cell, I have stated multiple times. I'm going to tell you here again. It's cell membrane. Yep. Very easy. How does cell membrane differ from cell wall? Now number one being, as I stated earlier, cell wall is made up of what? Cellulose. Whereas cell membrane is made up of lipids, right? But this is probably not the appropriate answer they're asking for right now, because they have not stated what the cell membrane is exactly made up of. But this could be one of the right answers to it. It's all right. But the appropriate answer which you could use here is the cell membrane. It controls what enters the cell and what leaves the cell, whereas the cell wall does not do that. And the function of the cell wall was what? To prevent cell from bursting. So that was its main function. It had prevented the cell from bursting, and whereas cell membrane controlled what entered and left the cell. So these are the main things that you need to know. And one more thing, the cell wall is of course thicker than the cell membrane, and the cell membrane is much more of a thinner layer. So these are the main thing that you need to know about, at least till here. This is the first part. I will make a second part regarding bacterial cell, and then we will do specialized cells, and that would be fun too in the next video. So hope to catch you in the next video. Hope you understood each and every bit of this concepts, and thank you. Allah Hafiz.